Hello, everyone. My name is Devesh Pangatabula, and I'm part of the HOPE Lab at the Department of Microbiology and Immunology at Drexel's University of College of Medicine. My project involves with pancreatic cancer in the development and aiding of novel reagents that aid in anti-tumor CD8 T-cell responses. Over the past couple of years, there have been many advancements in cancer research, such as immune checkpoint blockade therapy, where they sort of highlighted and differentiated the roles of T cells. In our project, we specifically look at CD8 T cells, and we've learned that they help in recognizing and eliminating infected or cancerous cells. However, despite these CD8 cells or the protection that is offered by our immune system, cancer cells can still find a way to develop mechanisms to evade or suppress these immune responses. And so our research helps to uncover and enhance these anti-tumor activity of immune cells, and we want to overcome the barriers that tumors impose in our microenvironment. More specifically, in our project, we focused on three different ideas: transformation, transfection, and transduction. Transformation is the process where bacteria uptakes extracellular genetic material such as plasmids. And in our project specifically, we use three different plasmids: T4, B4, and 4. And all these plasmids have this GFP in them, and GFP just stands for green fluorescence protein. And the whole reason for this is when we run these successfully transduced cell lines in a flow cytometer, we should expect GFP expression. And that just simply happens where we shine a laser with the machine into our samples, and it lights up. So getting back, another component is transfection, and transfection is the process where we artificially introduce our genetic material into our cells, chemically or via physical methods. In our project, we use platinum E cells, and I forgot to mention this before, but we also use stable 3s as our chemical component E. coli. And using these platinum E cells, we're able to develop our retroviruses, where we can later, in the process of transduction, retrovirally transduce our cells to express ovapeptides. Ovapeptides overall has a very high importance towards T cell activation. Our plasmids have varying level of expression when it comes to ovapeptides. So for future studies, we could study the activation expressions of different plasmids, but that's more for future validation studies, etc. So when we take a look at our data and results, left-hand side is our controls. It's going to be the same for figure 1 and 2. And our right-hand side are the ones that have been experimentally transformed, transfected, and transduced. In general, for cells that do not have GFP, we should be expecting a single shared population between all 4 graphs in each of the figures. However, if we do have GFP expression, which we want on our graphs on the right-hand side, because they are the ones that have been transfected and transduced, we should be seeing a separate distinct population. And that's clearly what we can see through our graphs when we've ran this data through the flow cytometer. And this just overall proves as a readout of success that our cell lines have been successfully transduced with GFP, and the ones that aren't don't have it. So for future work, our future validation studies involves doing in vitro studies of T cell activation or studying T cell mediated cytotoxicity of tumor cells. More specifically, the very next step in this process is to accomplish FACS sorting, where we'll successfully select only GFP expressing cells. We don't want the control ones anymore, so we'll just get the fully expressed GFP ones, and we could later use these cells to go culture T cells with tumor cells and use T cell activation markers and evaluate that with CD8, CD25, and CD69, and tumor death as a final readout of success. Overall, our development in these novel reagents will help aid us in our overall understanding and goal of studying T cell responses and T cell differentiation and T cell exhaustion in pancreatic tumor immune microenvironments. Thank you so much.